So yesterday, let me share my screen. Here it is. What and all we discussed yesterday, I think if I'm not wrong, the last thing that we were discussed yesterday was about the clinical trial protocol. Am I correct? I told you that a clinical trial protocol is the one which I can call it as an entry point for your clinical trial. Correct? So this is the first document, or it is the first one which will define your clinical trial. So this document was prepared under supervision of your sponsor. Sponsor is nothing but this is the person who is going to provide the funding for your entire project. I mean to say that for the entire clinical trial, sponsor is the guy who is going to provide the funding. But we also discussed yesterday that sponsor alone cannot do this particular one. Sponsor will take the help from either clinician, we can also call him as an investigator. Along with the investigator, we also have a biostatistician, correct? This is what I have mentioned yesterday, correct? So we have a biostatistician and investigator. Investigator or clinician are nothing but the doctor, right? There is a minor difference. The minor difference is going to be a clinician is the one who personally take care of the patient or the subject. The investigator is the one who will carry out the entire, right? Sometimes you may also have another role. We can also call them as a principal investigator. A principal investigator is the one, you can simply call him as a senior doctor, right? That's a basic difference. Okay. Sometimes the sponsor may not be an individual. Instead, we have CRO. What is CRO, guys? Anyone from yesterday's class? If you remember anything, what is CRO?
Clinical Research Organization.
Clinical research, no.
It's contract research organization.
Contract?
Research organization.
Exactly. It is a contract research organization, right? So when the sponsor is not an individual, in that particular case, we may also go for the CRO. The CRO is nothing but the contract research organization. Either of these two guys is going to produce the protocol. Remember, being the SAS developers or being the SAS programmers, right? You don't need to have an in-depth knowledge about the protocol. Doesn't require. Why? If you are part of a CDM, clinical data management team, or if you are part of your clinical research team, in that particular case, it is mandatory to know each and every piece of your protocol, right? It is. I'm not restricting you to learn about the protocol. I will guide that, and I'll also suggest that learn about the protocol, but don't go deeper into the particular one, because the protocol will be having around 14 to 15 different elements there. All the elements may not be required for us being a SAS programmer, right? Then again, the second reason why I say that don't go in deep about this particular means each protocol is different. From company to company, mean to say that from sponsor to sponsor, the protocol will be different. But there is a standard guidelines, right? So that is nothing but how a good protocol can be. There are guidelines for that. Every company or every sponsor or CRO more or less will follow the particular guideline. Sometimes there are violations as well, but we need to ensure that we means FDA will ensure that the guidelines are within the boundaries. All right. So I told you yesterday, or I have also shown you the protocol template. You can see that this is the protocol template. You can see there are approximately 17 different parameters here. 17, right? Going through each one of this, understanding each one of this is really a difficult thing, right? Every one of us doesn't need to do that. But whatever the basic information, especially for the interview purpose, we need to understand. All right. So today we spend some time on this particular protocol, right? And again, parallelly, we also try to discuss about, that is, first topic for today's class is going to be how the protocol looks like, what are the elements of the protocol. If you completely go with only the protocol elements, it looks like a very dry session, right? Completely a dry session. You may feel bored or you may feel exhausted. So parallelly, I'll also talk about crossover teams. What are the various teams we collaborate? We work with. Being a SAS programmer, as part of your clinical science, clinical SAS, you're going to collaborate with multiple teams. There are multiple roles will be there. Who and all? What are their responsibilities? That also we need to discuss. So parallelly, I'll also start this today only. In addition to that, I'll also talk about what are the various definitions which are involved in your clinical trial. That is, before we start with SDTM, this is our core. SDTM, ADaM, and TLF. These three are the ones we need to concentrate. But before we go there, we need to understand few definitions. We need to understand what are the various teams we work with, and this entire clinical research, the entry point is protocol, so we need to understand the elements. So from today onwards, for the next two classes, we discuss these three things parallelly. No worries, these three are individual. Once you complete your definitions, you will be able to understand your protocol properly. That is the overall objective. So we start with the protocol, slowly we discuss the elements, parallelly we go with the crossover teams, and we also discuss about the definitions. Clear? All right. All right. So a clinical trial protocol, the elements are here. So what are the elements? There are 17 elements, but I have given you like the crux of all the elements here. Ideally, the first element is supposed to be the title, but I haven't keep the title here because every one of us know how the title looks like. So I'm going to explain the title directly, right? But I haven't keep that here. So there is a title, there is a background, study rationale, objective, study design. This one we need to understand in detail. What is that study design? Subject selection, study treatments, what are the various procedures, adverse experience reporting. We can also call them as adverse events. Very, very, very important one. So discontinuation of the subjects, under what cases the discontinuation of subjects will happen. And also we need to talk about, I told you about the violations, correct? There are some violations will happen in the protocol. And I also told you that those violations should be within the limits. So what are those violations? And statistical considerations, that is nothing but what will be the, there is going to be something called efficacy. Efficacy is nothing but the efficiency. So what might be the efficiency? For few people, efficiency is 100%. For few people, efficiency can be anything about 80% also. What are going to be the considerations? What is going to be the data collection and monitoring? Make sure all the data that you are collecting is going to be ethical and regulatory considerations you need to continue with. We are going to discuss all of them starting with the title. All right. So let's start with the title. How a title looks like. So you can see that a title page is the one which can have the details about what is the protocol name, who is the sponsor, when this got started, that is start date, version date, what is the product that we are actually investigating, what is the development phase. You might be asking, Murli, what is this IND number? This IND number is also related to us, but we don't go in deeper about that. But the development phase is important. Meaning of the development phase is phase 1, phase 2, phase 3, phase 4 will be there, correct? At what level you are writing this? At what level you are writing it? Sometimes you can see here on the right hand side, that's the reason I have given you the example also. Can you see that on the right hand side? The phase development, here it is. The phase development. It is the phase development is 3. So it is actually representing the third phase of the development. So on the right hand side, whatever the example that I'm trying to give here, this example, this particular protocol is the protocol that we are giving for the phase 3, right? You can ask me, Murali, you said that it is the entry point for the clinical research, correct? So what is the meaning then? The phase 1 development, phase 2 development were already completed. For those two, phase 1 and phase 2, there were different protocols already, correct? If I'm continuing with the phase 1 itself, let's say all the 4 phases I want to include in the one protocol, I can do that. If not, I want to go with the different phases. For each phase separately, I want to provide the this one. In that particular case also we can do it. So it's up to us. Means that it's up to the sponsor. It's up to the CRO. Clear? Now let's see that how the title looks like. Can you see this particular title? A phase 3, because we're starting with the phase 3 development. Double-blinded, placebo-controlled, randomized trial to determine the efficacy and safety of a low and high dose of, this is the drug name, NS0314D as add-on therapy in subjects with Alzheimer's disease. This is a Alzheimer's disease treated with a stable dosage of single, what is this? Dopamine, not able to, maybe I need to zoom it. Agonist. Okay, first I'll tell you what and all we need to understand from this. Phase 3 we know, right? This is nothing but the phase 3. You can see the double blinded. So there are 3 types of blindings. Blinding is nothing but masking. Anyways, I'm going to discuss that as part of these definitions. So just I'm going to give you a overview. So there is going to be a single blind, a double blind, as well as the triple blind. Totally 3 kinds of blinds will be there. All the 3 blinds we are going to discuss. A protocol or the treatment can be a single blind, or it can be double, or it can be triple. Then placebo control. Placebo is nothing but a kind of a drug only, right? So what is the randomization? Like how many subjects we are taking? All these things we are going to discuss. Okay. So for now, we just understand that there is a title talking about some of the definitions and parameters. Those parameters were part of your definitions. Today itself, we are going to discuss about this blinding thing. Right. Background. Because if you see that the second element that we are going to talk here is going to be background. So what is the background of this particular one? Under the background, 4 things we are going to discuss, or 4 things we are going to concentrate. What is the drug composition? Right? Whatever the drug that we are going to take it, what is the composition of that? Mean to say that most of the drugs are not a single medicine, are not a single chemical composition. There are going to be more than one composition will be there. Best example is going to be, if you consider your one of the most common irregular thing in the Indian community is diabetes. Correct? We cannot call diabetes as a disease. It is a irregularity. Correct? So because of the food habits or because of the your living methods or your lifestyle, you are going to get this irregularity. Correct? So to manage this, what we are doing, we are consuming the tablets. So what is the most common tablet that we are going to use? There are going to be a lot of vendors, but the common composition is going to be, you are going to have the metformin plus there will be another most of the times. But the common composition is going to be metformin plus something else. Correct? So what is that particular composition? Right. Second, mention the disease or condition for which the investigational drug product is being evaluated. That is, whatever the drug that you want to take or that you want to consider, for what disease you are actually doing this, because metformin has multiple things. It is going to cure or it is going to act like a supplement for the multiple diseases. But for what disease, to treat what disease you are actually investigating this. Then provide a brief summary of non-clinical data that has clinical significance. We are going ahead with the clinical trials. There is some non-clinical data. Those are not falls under clinical, but those are important for our clinical trials. So if you have anything like that, brief them. Brief them. The last one is going to be provide a brief summary of the clinical things, clinical significance. That is nothing but what we need to test on all. That is called clinical. Other than the clinical, what is the other details which are having impact on our trial is going to be a non-clinical data. Both of them are required for what we are developing this particular drug. And finally, what is the conclusion? This is the basic background of your clinical trial. Am I correct or not? This is the basic thing. What is the medicine? What is the composition? To treat what disease we are doing this? What was the non-clinical data that you need to consider? What is going to be the clinical summary? Then, study rationale. Study rationale primarily talk about one thing: risk or benefit assessment. What do you mean by risk or benefit assessment? Every drug has a risk involved with that. Every drug we are taking to get some benefit. Am I correct or not? I'm taking metformin. Metformin we are taking to get the benefit of lowering your sugar levels. Very straight thing. The benefit is going to be, it is going to lower your sugar levels. But what is the risk involved there? I don't know how many of you are aware of that. Metformin is one of the primary reasons for cancer. It will activate the cancer cells, right? Though we all aware of that particular one, but still we consume the diabetic tablets because there is no other alternative. Either you need to control that by having a proper food, maintaining the lifestyle. In addition to that, since the sugar levels are very high, we must take this particular medicine. So the risk involved there is going to be, there is a high chance that it may activate the cancer cells, because every one of us will have the cancer cell. When it got activated, then the case is going to be different. Correct? So we need to understand how the specific risk of this particular product will be mitigated in the study. So we know that the risk is involved already. The risk is going to be, it is going to activate the cancer cells. I told you in the beginning, we always take this metformin plus another chemical composition. The plus another chemical composition is to mitigate your risk. That is nothing but it trying to suppress those cancer cells. Metformin is going to activate it because it is having two things. The benefit is going to be it lower the sugar levels. The risk is going to it activate the cancer cells. On the other side, the other composition that we are providing along with your metformin is going to lower your sugar levels. Sorry, it's going to suppress your cancer activity cells. Correct? So we need to understand this particular one, because if you are not able to mitigate the risk, it is not safe enough. If you remember, the primary reason of carrying out the clinical trials is to understand and measure the safety of the individual or safety of the subject. When the safety features are not meeting, when the safety parameters are not able to meet, that there is no way, or that there is no need of doing the clinical trials, or there is no need of developing your drug. Am I correct? So what we need to do? Assess your risk, assess your benefit. When you assess the risk, understand how to mitigate the risk as well. Is that clear? Right? Because we all know that it is to describe why investigational drug will be safe and effective for the clinical indication intended. Meaning, for what we are targeting, for what purpose we are actually taking this particular drug, that drug need to be safe and need to be effective. Right? If there is any risk involved, analyze the risk, assess the risk, and mitigate the risk. Then only we can proceed further. That's the reason the study rationale is very, 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 very important. Can I have a quick confirmation from everyone? 3 parameters we have discussed today. The 3 parameters that we have discussed here are: the first one is going to be under the elements, the title. Basically, the title gives the complete information about the health. It has the sponsor information, it has the version details, it has what treatment we are actually doing. Correct? and it also has the information about the primary investigator and secondary investigator and any other teams involved there. The team information will be available. Number 2, background. The background is going to talk about 4 parameters. What are the 4 parameters it is going to talk? It will talk about the drug composition. Second, it will talk about if I'm taking this particular drug to treat what disease it is going to be, we are going to consume this particular drug. So it is also going to have the details about the disease. Right. Then, what is the non-clinical data that have impact? What is the clinical data that we need to consider? That is about the background. Third thing, what is the third thing? The third thing is going to be, we are going to talk about the study rationale. So what is the study rationale talk about? It primarily talk about 2 things: risk, benefit. Obviously, we look at the benefit. The benefit is nothing but it primarily talk about the why I'm taking this particular medicine. If I take this particular medicine, what is the benefit I'm going to get out of it? But along with the benefit, what is the risk? Analyze the risk, assess the risk, mitigate the risk. Yeah. So 3 parameters, all of you are comfortable. Can I have a quick confirmation? Every one of you? Any questions here? Wonderful. Right. So the next thing that I'm going to talk here is study objectives, because we also have the study objectives, right? So now to understand this better, parallelly, we also see this particular document. Let's say this is one of the protocol. I told you yesterday I'm going to give you some of the protocols, right? So this is one of the protocol. Let me also open this particular. So rational, maybe I can also share this particular one after this particular class. I'll share this. This is the protocol for you. Can you see this? This is the protocol. This is 2.0 version. This is primarily for the drug NS0314D, the same that I have also mentioned in your this one also. Can you see this? Here, NS0314D. This is the investigational drug. For this investigational drug, this is going to be the protocol. All right. This drug is primarily to treat Alzheimer's disease. Okay. How it looks like. Title, we already discussed the title there because the title is already there. Now we are talking about the objectives. Can you see that? We are actually at the objectives page. This is the objective. So there are 2 objectives: primary objective. The second one is going to be secondary objective. For primary objective, what is the efficacy endpoint? Efficacy endpoint is nothing but this is the way we are going to measure the efficiency. You can understand in that particular way. This is the highest parameter for identifying at the same time for measuring your efficiency. Let's talk about the objective first, then we talk about the efficacy endpoints. Can you see the primary objective? The primary objective is nothing but what is the specific aim and objective of this particular trial, right? Why I am carrying out this particular trial, right? So to do that particular one, I need the data. So we are going to collect the data, right? We are going to collect the data to address a specific aim and objective of the data or of the clinical trial. Clear? Let's see how the objective is defined here. The objective of this trial are to evaluate the safety and efficacy of 2 doses of 0314D drug, NS0314D drug. One is 50 mg, another one is 100 mg, right? Compared to placebo as add-on therapy in the subjects with Alzheimer's disease, right? So what they are doing is they are currently receiving this one stable dose of single dopamine agonist. This is the single dose they are actually taking already. They are currently on this particular one. But what we have to do now, we want to test how safe it is and what is the efficiency of it sorry, we want to understand how safe and how efficient it is if I give two different types of drugs. One is going to be 50 mg and another one is going to be 100 mg. That is the primary objective, correct? So that is the objective. Here you can see primary as well as the secondary. So for the primary and for the secondary, what I need to do. So I need to evaluate the changes, right, from the baseline to W24 in a motor symptoms. So this is too deeper and this is too much related only to this particular Alzheimer's disease. So there is going to be a baseline. From the baseline to W24, till that we are going to evaluate. Clear? So second one, the secondary objective is going to be, this is UADRS section 3, this is another parameter. Don't go that deeper into that. Second one is, again I'm going to do the same thing, but here instead of motor symptoms, I'm going to do the same in the daily living, cognition, changes in the global clinical status, all these other things. This is nothing but a regular life. This is at the motor symptoms, right? So each drug has a specific target. So each protocol is going to be different. When it comes to us, when it comes to that being a clinical SAS programmer, we don't worry too much about it, right? But if you are a clinical data management guy, a clinical research guy, then you need to understand what is W24, what is called motor symptoms, what is called UADRS section 3, all these things. But for now, our objective is to understand the protocol. To understand the protocol, this information is more than sufficient. All of you are clear about it? Can I have a quick confirmation on primary and secondary objectives? It is all about objective is nothing but why I'm taking this, why I'm considering this particular drug, what I'm doing with this particular one. Right? Can I have a quick confirmation? All right. So it's going a little rough. So what we can do now is we can slowly move from protocol to, let's go to the crossover. So till now we discussed about the protocol, right? So now let's stop here because we talked about four different parameters: title, background, study rationale, and also we also discussed about your objectives, right? So let's stop here. The remaining 5 or 6 or remaining 5 or 10 we're going to discuss in the tomorrow session. For now, we can make a switch from here to crossover themes because until unless we change the topic, we don't get the fresh air. So let's change the topic and continue with the crossover themes. Again, tomorrow we will discuss with the remaining of them along with the remaining crossover themes and the definitions, like the previous. Okay, right. Now, let's come back to our slides. So, what are the various cross-functional teams that we are going to interact? So, with what people we are going to collaborate? So generally we have a sponsor, clinician or investigator. These two we are already aware of. The person who will provide the funding, the person who will carry out your investigation. So these two are we are pretty much okay and pretty much comfortable. That's the reason I'm not spending much time here. Instead, I'll be moving to the biostatistician. When it comes to the interviews or when it comes to the roles, there is always a confusion. The confusion is going to be, is statistician and clinical SAS programmer, are these two are the similar roles or the same roles, or are there any difference between them? Remember, biostatistician is entirely different from your clinical SAS programmer, right? A clinical SAS programmer over the years, by gaining enough knowledge with respect to the statistical terms, can turn as a biostatistician. There is a possibility. But ideally speaking, these two are two different roles. Right? Now you can see here clearly. The role of a biostatistician is to work with a clinician and clinical programmer. Remember this. Clinician is nothing but a doctor. A clinical programmer is nothing but you and me. Right? What are the responsibilities? First thing, help with the statistical design of the clinical trial study. So we need to understand what is called the statistical design. That will come under your definitions. Today only we are going to discuss that one as well. Then help with formulation of hypothesis. You guys remember where this hypothesis has come and where we discussed it? Anyone? If you remember the clinical trials procedure that I have explained on the day one, I told you three things. Number one, in each clinical trial, by end of each clinical trial, a question will be raised. A hypothesis will be developed. Third, analyze, assess, and repeat. Am I correct? First, a question will be raised. Second, a hypothesis will be created. Number three, analyze and assess the question and hypothesis, and then repeat the clinical trials if required. Remember that? So, simplest example. Let's go with a simple example. Let's say there is a person. Let's consider COVID itself. So there is a person who got COVID symptoms. He diagnosed with the COVID. Initially, let's say that during the drug development time or during the clinical trials, the investigator understands that. So let's say we have developed a drug. The drug has been given to this particular subject. The subject has taken the drug and he is recovered. Let's say that his body temperature becomes normal. He's not feeling any body pains. He almost feels normal. He doesn't have any COVID-related symptoms. So then investigator will say to the clinician, and investigator will update the information saying that our trial looks fine. By the time, the biostatistician along with the principal investigator raise a question. That is, what is the dosage that we have given? In how many days this guy has recovered? The subject has recovered. The question, the investigator can answer it. Then again, the principal investigator or biostatistician ask a question. What if, because we conducted this particular trial in a proper environment, controlled environment, meaning we have provided a proper oxygen to this particular guy, correct? We use the oxygen cylinder and we supplied the oxygen through the machine. Number two, we maintain the same temperature throughout the day because we are in an AC controlled room, correct? Number three, we ensure that there is no other bacteria or there is no other virus in this particular room because we're continuously sanitizing this particular clinical room or clinical site. So under these circumstances, the drug has worked or the vaccine has worked. What if the person is not in a controlled environment? That is, when the person joined this particular clinical research, let's say he joined in the phase 3, by the time he already have COVID, and we also understand that when we take the CT scan of his particular lung, we could see that he's already having, let's say, 15% of the lung already affected with the COVID. We can clearly see that particular volume in the CT scan. So we started giving him, first we started giving him the oxygen. Then he becomes stable. Then we started giving this particular drug. The hypothesis that I'm making here is, what if we provide the drug without using this particular oxygen support? What if we are not giving this particular drug in a controlled environment? What if we are not continuously sanitizing this? Under those scenarios also, will this drug work or not? That is the hypothesis or question that we gave. Right? Number 3, determine the optimal size required to guarantee the level of precision. That is nothing but, optimal sample size is nothing but how many number of people that I do or how many number of subjects that are required for this particular randomization. Give the sample, right? So give this particular sample to get the optimum precision. Calculate the appropriate sample size for the study to address the specific aims. Both of them are going to be randomizations. Give the proper samples to carry out your study properly, right? Determine the data variables to be measured and how they related to the study objectives, right? Let's say that when it comes to your COVID again, because I'm taking this example more frequently, let's go with the COVID. When we are going with the COVID, right, what are the vitals that are important to me? What are going to be the various parameters I need to measure, which are related to this particular COVID? I need to concentrate on that. What is the primary parameter? We need to check the temperatures. Second thing, we need to check the oxygen levels. Exactly, Sachi, right. We need to check the temperatures first. Second thing is going to be need to let the blood oxygen level, which is nothing but here is SpO2. Correct. So these two are the primary things. Later on, we'll take about the your blood pressure. Later on, we consider about the all other things. Correct. Let's say we are talking about the dengue. When I talk about the dengue, SpO2 is not my primary parameter. My primary parameter is going to be the platelets count. Correct. What is the platelet counts of this particular patient? Are the platelet counts going down or are they stable? These are the parameters for me. Correct. For each of this particular drug or for each of the particular disease, the relative parameters only we need to measure first. We need to give the priority to the relative parameters. So what are those relative parameters? Biostatistician is the guy who's going to help with that. One. Develop the data collection process, provide the method for analyzing the data, and finally interpret the results. Meaning, so we have the data coming from the various sources. So collect the data and cleanse the data, because the data can be data is not going to be proper all the time. The raw data is going to have a lot of things. You need to properly cleanse the data. So the data cleansing, the data collection will be taken care by the biostatistician. Only the process he will develop, but the original data cleaning will be done by the clinical SAS programmer. Second, what are the methods we need to provide? What are the methods for analyzing the data? The methods need to be provided by the statistician. Finally, how to interpret the results? Whatever the results we got, how to interpret them? All these things can be done by a biostatistician. Remember, biostatistician is the most critical guy in the project. I'll tell you why. If I ask you in the SAS, even in the SAS also, maybe it is not that relevant, but at least I can tell you. If someone asks you, hey, this is my project, my end outcome is going to be, I'm looking for all the people who suffered with COVID and who got recovered. Just two things: who suffered, who got recovered. That's it. Being a developer, I follow one method. We have 15 more people in this particular group. Each of us may go with the different objectives, or may go with the different methods, or 2 or 3 of us may follow the same one. Maybe simple example. If I ask you, hey, can you give me how many number of observations in a data set? This is the question that I ask. 2 or 3 of us will simply go ahead and will say that, hey, I use the proc SQL and I calculate the distance count. Someone else will say that I don't use proc SQL. Instead, I'll go with the data step. In the data step, I'm using the n option and I'll calculate it. I feel it is more efficient. Someone will say that I don't even go with that particular one. I'll see what is the maximum value of underscore n underscore, and based on that, I'll calculate. Correct? Someone will say that I don't even go with all this. I simply go with the proc frequency. The proc frequency, whatever is going to be the cumulative number, that number I'll take it as my outcome. So 4 different methods, right? But the end is going to be the same. The end result is going to be the same. Each one of us is going with a different procedure. Same goes with the biostatistician as well. Biostatisticians doesn't have a standard approach for the given solution. Let's say you and me, both of us are 2 different biostatisticians. My approach is entirely different from your approach. So when the project started, I have provided one approach to you. So the investigator started the process and it is going on. I entered into the phase 4. When I entered into the phase 4, I decided that I don't want to work with this particular company. I left the company. So a new biostatistician has appointed. But the new biostatistician, there is a very minimal chance that he will continue with what is left by the previous biostatistician, because it is very difficult to understand what is the approach he has used. So what is going to happen? The moment I taken the charge of biostatistician, I will give my own approach. When I give my own approach, what is going to happen? Everything is going to change. So whatever the previous approach was there, that completely lost. I don't continue with that particular one. I will go with a new approach now. Right? This will increase, or this will make delay in the deadline, because whatever was done till now is gone. Is gone. I need to start freshly from here. I will change the random variables. I'll change the population. I'll change the deliverables. I'll also change the analysis methods, and I also change how I'm going to implement that. So the biostatistician is the one who is very crucial. Because of biostatistician, most of the time the deadlines will change. Clear? Right. The next, data management team. We can also call it as a clinical data management team, which will be led by clinical data manager. So a clinical data manager is the one, right, who is primarily responsible for case report form. I'm going to show you how it looks like, maybe, but not today. In the next class, I'll give you a simple CRF, case report form. He is the one who is responsible for your database design. He is responsible for your data entry, the validation and discrepancy management, medical coding, data extraction, and database logging. You can see that all these are almost related to us. You need to say that from here. Correct? Right? They will design the database, they do the data entry, they do the validations, they also do your medical coding, and they also complete your data extraction, and they log the database. The moment the database is logged, from there, clinical SAS programmer can take the charge. But on a whole, a data management guy is the one who will be primarily taking care of the CRF. Right? There is a normal CRF and eCRF. I'll give you the sample template of this and how a CRF looks like in the next class. All right. In addition to that, we also discuss about these and all common terms. You can pretty much understand this by this time. What is called database design, what is called data entry, what is going to be the data validation and discrepancy management, what is called your medical coding. Medical coding is not related to us, and how we are going to extract the data and database. All these are part of your data management. Medical writer, a very crucial role, because this is the guy, whatever the outputs that are created by the SAS programmer, being a SAS programmer, we are going to create a table or a listing or a figures. Correct? We prepare this and we'll give it. But the person who is on the other side, that is the people who are going to review your data, the people who are in the FDA, they are not the SAS programmers. Correct? They don't know how to read it. Correct? So what they need to do now, since they are not aware of this particular one, since they are not aware of this particular how to utilize the SAS tables, listings, and figures, then this particular medical writer comes to the picture. This guy who is going to produce a scientific document, and that scientific document will be viewed and used for the entire research. Right? This is the guy who is going to utilize the tables, listings, figures created by us and prepare a meaningful scientific document. Right? So we need to understand to whom and all he will collaborate. A medical writer, he will work with doctors, he will work with the scientists, he will work with the SMEs, subject matter experts, and then he is going to create a clear research result, product use, and the medical information. all the things. This document is the final document, right? And also he need to ensure that it is not a very easy thing, because whatever the document he is preparing based on the tables, listings, and figures need to be in compliance with the regulatories. Either it can be EMA, or it can be FDA, or it is going to be our drug control, whatever it is, whatever the guidelines that they have provided, the document that is going to produce should be in compliance with this particular part, right? And also, while he preparing this particular one, he closely worked with your biostatistician. He closely worked with the SAS programmers, because SAS programmer is going to develop this, but the input for the SAS programmer will be coming from the biostatistician. So a medical writer who is going to consume our output will frequently in touch with the SAS programmers as well as with the biostatisticians for better understanding of the results. Can I have a quick confirmation? All of you are able to understand this? Is this clear? What are the cross-functional roles, or what, to whom and all we are going to collaborate? So what are the cross-functional teams that we are going to work on? Sponsor. Maybe we don't get direct interaction with the sponsor. It is very hypothetical, very rare scenario you are going to get chance to work with the sponsor. Second one is going to be the investigator, or we can also call it as a clinician. That is the person who is going to take care of the patients or subjects, or who will carry out your clinical research. The third thing is going to be your biostatistician. A biostatistician is the one, primarily speaking, he is the one who is going to provide the logic to us. He is the one who is going to say, who is going to provide the algorithms to us. He is the one who is going to give the details about what is going, how the data looks like, and what are the various analyzing patterns that I need to follow. We can look from only from the SAS perspective. In that case, I can say these three. He will also help us to validate the data. Then there is going to be a clinical data management team. There is a manager who will be taking care of the preparing the case report form, right? And also he will be responsible for the database design, data entry, data cleansing, and also database. And then we have someone called medical writer. The medical writer is the one who is going to utilize the outputs that are produced by the SAS programmers. Those can be tables, those can be your listings, or those can be figures. He will utilize those particular things, and he will be in compliance with the regulatory guidelines, and he will prepare a scientific document. That scientific document is going to be the final document which will be produced to the regulatory authorities. While preparing this particular document, medical writer closely work with the SAS programmers and also with the biostatisticians. Is that clear, guys? All of you? Right. So can I continue with the definitions, or can I stop here? It's up to you. Can I go with some of the definitions, or we can pause it here and we can continue tomorrow? It's up to you guys. Anyone? Can we do it tomorrow? Subhasri? Okay, fine. Okay, let me set up the base for tomorrow. Tomorrow, guys. Tomorrow we have the class. I'll go with the class tomorrow also. So I had some plans to go to my native. That's the reason the day before yesterday I told you that Monday and Tuesday I'll go to the class, and I don't take the class for the next 2 days. But tomorrow also we have the class same time, 8 p.m. Okay. So what are going to be the things that we are going to discuss tomorrow? Let's set up the base. We will continue with the remaining elements. At the same time, we will understand some of the definitions. The definitions include, let me write it here. First, we will go with the study design, the most important one. There are two study designs. Number one, parallel study. Number two, crossover study. So what is this parallel study and crossover study? We are going to discuss. Number 2, once we complete this, we will go with the blinding. Blinding is nothing but masking. So there are 3 types of blindings: single blinding, double blinding, triple blinding. All 3 we are going to discuss with simple examples. And then we need to understand what is called sign. That is very straightforward only, but we need to understand what is called sign. In addition to that, there are few more. I can say that what is called multi-center study, what is called multinational study. We also talk about what is called unblinding. Sometimes we go with unblinding also, no blinding at all. We will discuss that. And then we talk about ICF screening and evaluation. So maximum how much time we can spend there, we can spend on the definitions. Definitions and protocol is done means we can happily enter into the testing. Right? So I expect or I believe that by tomorrow I can complete these definitions so that we can start with the testing. So this is the objective for the tomorrow's class. Clear, everyone? All
Sure, hi, sir.
That's all. Yeah.
Yes, sir. Can you share me this PDFs everything?
Which one? Yeah, I will share it with Bolusar today itself, right? So just have a word
Okay.
with Bolusar, and so I'll share the protocol, I'll share the template, and also I'll share the slides. This one.
Okay.
All these I'll share. Okay.
Yeah. Thank you.
Thank you.